My name is Tom Wolfe. I'm a PhD candidate in Dr. Frederic Chain's lab, and I'm going to briefly talk to you about our research into the role of epigenetics in the evolution of gene duplications. Along with mutations of individual nucleotides, a common source of new functional genetic material is the phenomena of gene duplication. As an important example, a series of duplications in the vertebrate lineage, circled here in orange, led to the existence of multiple globin gene varieties, which play an important role in oxygen transport and storage. However, not all duplications are beneficial. In fact, most are either neutral or harmful. To illustrate how an extra copy of a gene can be harmful to an organism, I'll take the example of a gene that produces a member of a protein complex. With a single copy of this gene expressed, you get the normal amount of its protein product. This amount has been selected for to exist in balance with its binding partner, and their co-expression in the right amount leads to a functional partnership. However, if such a gene is duplicated, then it will potentially express twice the normal amount of the protein it codes for. If this occurs, it may not simply be that the excess protein is unused, but that it less often binds with the necessary amount of its partner, leading to incomplete and dysfunctional protein complexes. There are several other ways in which an increase in the amount of a gene might be deleterious, but despite this, duplications that might be expected to cause such an imbalance nevertheless exist, and so naturally the question is how. One proposed explanation is that to avoid dosage imbalance, the expression of duplicates might be reduced by what are known as epigenetic modifications around the time of duplication. While some of these modifications can increase the expression of genes by opening up the genome to the gene expression machinery, they can also restrict access to genes or prevent the gene expression machinery from binding on the chromosome near that target gene. Supporting the hypothesis that these modifications reduce duplicate expression, it has been shown in several studies that evolutionarily younger duplicates have higher levels of repressive modifications that decrease as they diverge, and lower levels of activating modification that increase as they diverge. In our own research in the genome of the social amoeba Dictyostelium, we have found evidence of decreased activating modifications at a critical gene location just upstream of the start of young gene duplicates when we compare them to older single-copy genes. We have also identified a lower level of expression among young duplicates, consistent with the idea that they are repressed shortly after duplication. To continue our research into this hypothesis, we plan to expand our analysis to members of the hominid lineage, a group that has had a large increase in gene duplications in recent evolutionary history. Using epigenetic sequencing data from these organisms, we will investigate how much regulation of young duplicates followed the pattern described before in this lineage, and whether epigenetic repression might precede duplication events or help to predict their occurrence. Thank you for taking the time to watch this video.